how this information are decoded or coded. So RNA is probably the one which has functioned in the early age, in the early phase of evolution, and the genes of all the modern cells are, and many viruses are made of DNA. So what I want to refer to is that DNA is the basic genetic material. Though earlier it was thought that DNA is the only genetic material, but later on it was found that not only DNA, some of the RNA molecule also carry genetic information. As you have learned that in some viruses, the RNA acts as a genetic material. That part we will talk a little later. Then what are the genes? You might have learned about the genes, you must have learned about the DNA, you know the chromosomes. So when we talk about the genes, genes are very specific sequence present in the DNA molecule that specify the kind of a protein they are going to make. So the protein, they are made up of amino acid, and this amino acid sequence comes from the information which is coded in the genes. And the templates for protein synthesis are not DNA. So you look at the flow of information, how the flow of information occurs from nucleic acid to the protein. If I look at the structure of the DNA has certain information. It passes on the information to RNA, and RNA passes the information to the protein. And protein is the one which functions. So what you have here, the DNA has a very interesting role where it transcribes RNA. So whatever the information is present in DNA is given to the RNA, and the RNA, whatever the information or code present in RNA, is passed on to the protein. These are complicated process, and individual aspect you will be learning in more detail. Not here, some information you get here, but most of the information you will get when you learn about the genetics and molecular biology. Here, our purpose is to show that how DNA can transcribe RNA, so how it is, converted. RNA can form protein, give you some basic information. And you can look at the bottom of this. I have given you some very interesting point, which is called as a central dogma. So what this central dogma means is that the information is earlier thought that it will move only from DNA to RNA and RNA to proteins. But there is an exception. Now we have learned that the information, the one which I am talking about, DNA to RNA, could be a flow from RNA to DNA. So what should we call it when I convert RNA into DNA?
Reverse transcription.
It should be the reverse transcription. And this happened because of the Nobel Prize winning investigation, which has led that RNA to DNA conversion is a possible because of the enzyme which is present in certain viruses. And this is known as a reverse transcriptase. Now let us move on to more details. So what you have learned so far is that DNA is a mostly a hereditary molecule, and nearly all cellular life forms, including viruses, they have this as a genetic information. Of course, as I said, the exception can happen in some of the viruses where you have RNA as a genetic material or hereditary material. Now, looking at the function of the DNA has two major functions. One is that it directs its own replication during cell division, and the other one is that DNA transcribes and it forms a complementary molecule, which is RNA molecule. So what you have role of DNA? On one hand, DNA can form another DNA molecule. That means duplication. So that way, the information can be transferred from one generation to another generation. The same copy of the DNA can move from one generation to another generation. Now coming to the another role is a transcription, which you have learned in the previous one. So what is the transcription? If I make DNA to DNA, I call it duplication or replication process. But if I use So this is the DNA. If I make another DNA molecule from here, this process will be called as a duplication process or replication. And this is what is when within the cell, if I convert DNA into RNA, this is known as a transcription. That is, it's transcribing its message to RNA. And that is the RNA which actually is responsible for synthesizing or giving the message to the protein molecule. So this part is elaborated later stage as well. Now, so you have learned about the function of the DNA. Looking at the RNA, RNA has a varied biological function. Its biological functions are different because there are different types of RNA. So one of the most common type of RNA is a messenger RNA. So what messenger RNA does? Messenger RNA is the one, as name indicates, carries the message. The messenger RNA can carry message from DNA, and this message will be helpful in synthesizing proteins. So these are information carrying intermediates in the protein synthesis. So if I say that DNA can synthesize RNA, so you have a DNA molecule, this DNA molecule can synthesize mRNA. So this mRNA, it can synthesize other RNA as well, but the mRNA, which I'm talking about, this mRNA is the one which carries the message, and this is the one which moves to the protein. Now, coming to the another, type of RNA, which is a transfer RNA. So this transfer RNA is the one which is carrying the message as well as they carry the amino acid. So actually, when the protein synthesis occurs, where amino acid has to assemble, form a protein structure, this will be happening on the surface of mRNA. So you have mRNA. So if I say that suppose I have a point where the assembly occurs. So you have ribosomes. On this ribosomal structure, you have mRNA, and at this mRNA, your tRNA will come, and then the recognition between mRNA and tRNA will be demonstrated by presence of codons, which is present in mRNA, and anticodons, which is present in tRNA. And here you have amino acids. So you have amino acid which is present in tRNA. So what will happen? If I know what is present as a message in the mRNA, the same kind of a tRNA which has anticodon for this mRNA will bind, and that is why it can leave amino acid here. Then another amino acid will be from the another code. So you have a codons present in mRNA, which will be recognized by the anticodons present in tRNA, and tRNA will have amino acid bound to it so that the amino acid can assemble near each other. And then, of course, there is energy requirement and other things to allow the amino acid to assemble. And when amino acid assemble, what you get? You get proteins. So that's the where protein synthesis occurs. Now, coming to the third part of ribosomal RNA, it is a part of protein synthesizing machinery. So, ribosomal RNA is present in these structures which I have shown. These are called as a ribosome, and these material could be a structural material, could have a lot of information. So look at the information, how this information is being transferred. So you have a codons. They are sequence of three bases. So these codons, they are sequence of three bases, and these bases are the one which will have the information to be transferred for the the so you have a codon, it will have a sequence of three bases, and these three bases I have shown you in the messenger RNA. If it is present, what it can do is that it can specify which amino acid can bind. So tRNA molecule which is having anticodon, they will bind with mRNA, and that's the way message passes on. So codons in mRNA are read are read sequentially by tRNA. So if I have mRNA here, it has codons, so it will be in triplet. So these are the codons which are present in mRNA because of certain reasons to show you how it is being recognized. So this is your mRNA. So in this mRNA, what will happen? The tRNA molecule sequentially act as an adapter molecule during the process of synthesis. What does it mean? It means that tRNA molecule which contains anticodon, which can be recognized by these codons present in mRNA, they can assemble in a particular way. So what I mean to say is that suppose tRNA containing alanine amino acid is present in first one. Second one, if it is codon which is specifying anticodon for glycine. So once you have alanine, then you have a glycine, then you have the codon which is specifying for the binding of tRNA which carries the information for phenylalanine. So what you have alanine, glycine, and phenylalanine. So this way protein synthesis occurs. Now comes next question, protein synthesis, where does it take place? So as I said, it occurs on the surface of a ribosome. So this is your ribosome. I am drawing this structure. These are the ribosome. On these ribosome, your mRNA is bound, and to this mRNA, tRNA will come and recognize it, and then it will leave a particular type of amino acid. So it will have amino acid which will be allowed to bind. So these are the complex protein synthesis process which you will be learning when we talk about. I will not talk in my course, but later on you will be taught how the protein synthesis occurs. At this point of time, it is very important for you to just understand what are mRNA, what are tRNA, what are rRNA. So rRNA will be present in the ribosome, the structure which I have shown. Now this RNA will have RNA. It will have a lot of other protein. So as I said, that the protein synthesis takes place on the ribosome, which are complex assembly of rRNAs and more than 50 types of protein. So this is a complex protein structure, which is a kind of assembly which we call as a ribosome. So on the surface of ribosome, mRNA is going to bind. mRNA will having codons will give information that which anticodon should bind. So that if I say that there are several tRNA which are floating around carrying a particular amino acid, they will be recognized by mRNA, and that's the way your amino acid they will come together. So how the message is being passed? Can you tell me, any one of you, how the message is passed? You have a DNA. DNA has synthesized the mRNA, and this mRNA has information that which amino acid should bind, and these amino acid will form protein, and the protein will do its function. So as I said, the genetic information is flowing from the DNA to RNA to proteins. Now, interrupted character of most eukaryotic genes are mosaics of nucleic acid, and sequence are called introns and exons. What does it mean? It says that if I have a gene or if I have a DNA which has got different nucleotides, and what it does is that it has some information which is in the form of introns, some of the information which is in the form of exons. And it has been seen that both are but introns are cut out, introns are removed, and newly synthesized RNA molecule, leaving mature RNA molecule with continuous exon. So if I have exons which has got certain information, so basically what you can say is that the introns, they carry the genetic information, but it is not being translated. It is not going to be transferred from one generation to another generation. So some of you may ask why these introns are present. So introns are present because of the evolutionary process. So suppose there are introns, and if I change a little bit in the genetic information, the whole codon will change, and that's the way that in the can occur. So that is beyond your course. I'm not going to talk about. But what you need to understand here is that what are introns and what are exons. So these are the information present in terms of code, which can be finally transferred to the next generation. Now coming to the more interesting point, which is the structural information. So what are the DNA? By now you might have got some idea that what are DNA and RNA. One thing is very clear, that you understand both DNA and RNA. One is a deoxyribonucleic acid, another is a ribonucleic acid. Then both of them, they are the one which are responsible for information transfer. So DNA is carrying information, giving it to RNA. RNA finally passes on to the protein. Looking at the structure, they are DNA. So they are the polymeric structure. So what is the DNA? DNA is a deoxyribonucleic acid, and these ribonucleic acids, they are made up of polynucleotides. Now looking at what these polynucleotides are. These polynucleotides, monomeric unit will be nucleotides, and these nucleotides, as I said, will be made up of one sugar unit. Each nucleotide will have a sugar unit, will have a phosphate, and will have a base. So sugars to another phosphodiester bridge. So you have a phosphodiester bond. So the base, the backbone of these phosphodiester bridge is referred as a is the one which is a backbone. So you have a chain of sugar units and linked by phosphodiester bond. So they are the one which are called as the backbone of nucleic acid. Now looking at the DNA and RNA, both of them, they contain sugar, both of them, they contain base, both of them, they contain phosphate. But where they vary in two aspects. One, which is a sugar residue. So in case of a DNA, the sugar residue is going to be deoxyribose, whereas in RNA, it is going to be ribose sugar. So there is a one major difference between RNA and DNA, where DNA is deoxyribose, whereas RNA is a ribose sugar. Slowly you will learn the information that why DNA is having deoxy, whereas RNA is having ribose sugar. Then there is another difference in the major bases. So I'll talk about what is this difference between the base residue. So coming to the bases, there are four types of bases, and two of them, they are derived from purine. I'll talk about what is the difference and what are these purine bases. So these are adenine and guanine, and there are two pyrimidine bases, which is in case of RNA, it is cytosine and uracil. In case of a DNA, there is a one difference, that this uracil is modified. Hello? Yes. See, I'm in a class. Can we talk a little later? Yeah. Yeah. Sorry for interruption. So we are moving on. I thought that there is a call from you guys. Are you able to listen?
Yes, sir.
Okay. So what you have in a DNA molecule, if I look at the base structure of a DNA molecule, so what you have in the DNA is that you have a sugar residue, then you have a base, so you have a red color which is showing sugar, and then a base molecule, and they are linked by a phosphate bond, and this phosphate is referred as a phosphodiester bond. So you look at the structure, this whole structure is giving you a backbone. So you see red color and blue color, that is giving you a backbone structure, and the base, which is a variable, is in yellow color. So this is the linkage where one sugar is attached with the another sugar by a phosphate residue, and this is the phosphodiester bond. Then you have a different types of bases, base 1, base 2, base 3, base 4, and so on. So what is the backbone? Sugar, phosphate, sugar, phosphate. So this makes a backbone. And the bases are the variable. Now looking at the detailed structure, in the backbone, what you have is that, as I said, this is this is the one which is your sugar unit. This is a phosphate residue, again sugar unit, then phosphate residue, sugar unit, and phosphate residue. And you have then bases. These bases are linked with the sugar. Can you tell me how the base can be linked with the sugar residue? What could be the bond? Type See, one of them is a sugar and another is a base. Now, I slowly will learn the bases. They will have a nitrogen, oxygen, etc. So, if it is a linked by N or a nitrogen, we call it as an N-glycosidic linkage. So, you will see that how they are linked. So, in case of a DNA molecule, what you find is that this residue is deoxygenated, whereas in RNA you have a hydroxyl group. So in this hydroxyl group, if I remove one of the oxygen, it becomes hydrogen. It is very clear that presence of this hydroxyl group in RNA molecule makes them susceptible under the basic environment. So what will happen if I have another hydroxyl group? So if there is an excess of hydroxyl group, what will happen that this hydroxyl group can attack on this. And if they attack on this, what will happen? They will lead to the formation of a linkage between this phosphate residue this oxygen. So there is a kind of a cyclic nucleotide will be formed and this base will be cut. So actually, in case of a hydroxyl group or in place of the basic environment, the RNA is more susceptible to be degradation where the bases can be cleaved. Is it clear to you how the base can be removed from this?
Yes, sir.
There is an attack by hydroxyl group.
Sir, could you please explain it again?
See, this hydroxyl group, so if you have a basic environment, what will happen? The hydroxyl group will be there, excess of hydroxyl group. So these electrons, they will attack on this. So what will happen? This OH residue, this hydrogen will be removed from here. You have one oxygen, and this oxygen will form a bond with the phosphate residue. And this bond, this phosphodiester bond will be broken. So actually what you'll have is that you will be having cyclic nucleotide. So this will get cyclized and you will be having cyclic nucleotide. And this way your chain will be broken. Whereas here, if I have a hydroxyl group, it does not attack on the hydrogen because there is nothing, it cannot cleave. So it becomes resistant. And that is why DNA is more stable in case of your, alkaline environment. And that is where it is thought that probably there has been evolution. It is thought that probably all the molecules were the RNA molecule. But in order to give the stability, it got deoxygenated at the second position. So this is the second position where deoxygenation occurs. And instead of OH, it has become deoxy group. Okay, so let us move on. Now clear?
Yes, sir.
Okay. Now let us move on. What are the different difference and how they are present? So there are two difference. One is in case of RNA molecule. If I talk about the RNA molecule, what you have is in RNA, you have hydroxyl group. Rest structure is the same. So you have the same structure. This structure, this is a ribose 5-carbon compound. As you have learned that it will be having a, in the structure which is having here you have CH2OH, like you have learned in case of glucose molecule where that has ribose is a 5-carbon residue. It has got at the second position OH group, but and it is a part of the DNA molecule. But RNA molecule. But in case of DNA molecule, there's a difference. This oxygen is removed, and that is why we call them as a deoxyribose. So the deoxy term refers to removal of oxygen from here. Now, looking at another difference. Before I talk about the difference, let me tell you that there are two types of the base residues. One is a purine residue, another is a pyrimidine residue. If you look at the purine structure, it is a very interesting structure of the molecule, which probably has been talked to you earlier. And these are the parental molecules. So purine are nothing. It is a parental molecule. It's not present in the, I cannot say the same structure is present. These are the parent, which rise to two different types of purine residue. So one is adenine and second one is a guanine. So you look at the basic structure, it remains same. Both the structures are same. But what is the difference? If you look at the difference between purine and adenine, so this is the base molecule, which is going to parent molecule, which is giving rise to these two structure. And the difference here is that you have the same structure, but there is one amino group. So what should we call adenine? We can call adenine as a 6-amino purine. So you look at the structure. At the 6th position, if amino group is present, the purine is called adenine. Now, in case of a guanine, you look at the structure. What you find is that at these two positions, modification has occurred. So, second position, you have amino group, and sixth position, you have oxo group. So, guanine can be called as 2-amino 6-oxopurine. So these two are the derivatives of purine, and purine is a two-fused ring. It is a bigger structure. I'll talk about some of the significance of this also, that this is two rings fused together, and that is what gives you a purine structure. On the other hand, pyrimidine is a single ring structure. If I look at molecules, one is a cytosine and another is uracil. So this cytosine, if I talk about the cytosine, so what are the cytosine? If I say the pyrimidine is the base molecule, what are the places where the modification has occurred? There is one position here, another position is here. So this is called as a 4-amino 2-oxopyrimidine. So fourth position, there is the fourth position where the amino group has got attached, and so pyrimidine is the parental molecule and cytosine is 4-amino 2-oxopyrimidine. Look at the structure of a cytosine. What is a so we have talked about cytosine. Now looking at the structure of uracil. Can you tell me what is uracil? Now very easy. Where are the modification? These two positions.
2 and 4.
So what we can call it 2,4-dioxopyrimidine. So uracil is nothing but 2 and 4-dioxouracil. Now coming to the thymine. Thymine, you see the same structure like uracil. So this is structure and this structure. I'm just leaving one part of this structure. This is structure and this structure is same. Except that this is the fifth position where one methyl group has been attached. So what you find is that in case of thymine, the structure is where you have 5-methyl 2 and 4-dioxopyrimidine. Now let me tell you a very interesting point here. The uracil is present in RNA, whereas the thymine is present in DNA. So what you see is that between uracil to thymine, there is a process. What should we call it, this process? There is a methyl group which is
Methylation.
added. It's just a methylation. So when uracil is methylated, you get thymine. And when DNA synthesis occurs, so in the early molecule, you will have uracil, which is getting methylated, and that's where you get thymine formation. So you have learned about the difference between DNA and RNA. You have, and you have learned how they are being. That is there any advantage of having thymine in the DNA structure? Answer is yes. And why it is so is because thymine distinguishes the RNA and DNA in terms of their chemical composition, and that becomes a very important point for the stability of the DNA molecule. One thing you have learned, that DNA is more chemically stable than RNA molecule because of the presence of this deoxy group over here, and deoxy group is giving you. I have not drawn the structure, but if you look at the structure, the phosphate bond if cleaves, so you will have a structure something like this here, this part. So if I take this base structure, which is so if I look at the stability, what you have is that here you have HH, so you have H here, H here in the case of So I'm just talking about RNA, what kind of structure will happen. So when OH group attacks, the phosphate residue breaks down, and here what you have is you have a kind of a bond formed where this will be phosphorus here from phosphodiester linkage. One side you will be having oxygen negatively charged. Here you will have this oxygen. So this way you get, can you name this molecule? It is 2' 3' late monophosphate derivative. So what has happened? The chain has a broken, and you have the cyclic residue. And because of this cyclization, there is a strain in the bond. So the oxygen, which was supposed, this oxygen comes over here. So because of this bond formation, this cyclization, the bond between one residue and another residue is broken. And that's the way DNA, RNA chain is broken. But this kind of thing is not happening. Second point which I am going to discuss about the stability of the DNA molecule is because of the presence of thymine instead of uracil. Now question is that how we talk about that? There is a very simple process that what happens that generally there are something which is happening within the cell and continuously where this cytosine molecule is converted into uracil. Now, if I want to cytosine to uracil, what are the process? You have to deaminate it. So there is an amino group which should remove and one oxygen should attach to it. So this is a chemical process which occurs and it's a very extremely slowly process. And that is what happens in the DNA and RNA molecules. Susceptible to deamination reaction, and that is why they can form uracil. Now let us take two scenarios. Scenario number one, where deamination has occurred and some of the cytosine has got replaced by uracil. So this can happen, and this is happening even now in case of RNA. So if one residue in RNA is changed, probably this information is not being passed down from one generation to another generation. But if that happens in case of DNA, it will make a change. It will make a mutation, fast mutation. And that is why uracil has been replaced by thymine. So you may ask, featured in thymine. Actually, what happens, thymine as such is not helping or not giving a resistance, but what thymine can help you is that thymine helps in the recognition process. How? If thymine is present in the DNA structure, there is no problem. The duplication will happen. But suppose uracil is present and which is formed, uracil is present in the DNA, what will happen? What should happen? Because this is not normal constituent of the DNA, it will be removed. And this process by which the randomly formed uracil is removed from the DNA is known as a proofreading ability of the DNA. So DNA has a very interesting point, as you have learned, that if you do proofreading, what you do is that you don't write complete sentence. So it is not the complete transcription process or translation process. What you do is that you just remove one of the residue and replace it by the original residue. So in a structure where I need a thymine, instead of that, if uracil is present, what we will do is that we will remove and replace it by thymine. So actually, the presence of uracil in DNA will make a choice that this is not the one which is the native one. It is coming from somewhere outside, and that is why it should be removed. And that's why they are being removed from the system. So these two features makes DNA more stable chemically in the environment. Now coming to more details, the nucleotides are the monomeric unit of nucleic acid. It is recapitulation. A unit consisting of a base bonded to sugar is referred to nucleoside. So what is the difference between nucleic acid and nucleoside or nucleotide? Nucleic acids are polymeric unit. They are made up of nucleotides. And in nucleotides, if I this is slightly, just you have to learn some very basic thing and you can understand. So there is a term which I have given as a nucleotide, and there is a term which is given as a nucleoside. What it base is that unit consisting of a base and sugar. So what does it mean? The difference between nucleotide and nucleoside is that the nucleoside does not contain phosphate residue. So if I, so this is the bond which is between the base. So this is the base, this is the sugar, and here the phosphate group is missing. So if phosphate group is not present, we call them as a nucleoside. If I add a phosphate group here, this P double bond O and OH, this becomes nucleotide. The four nucleoside units of RNA are called, this is just for nomenclature, your understanding. They are called as adenosine, guanosine, cytidine, and uridine. And the in DNA are called, so RNA, they will be called as adenosine, whereas in DNA, what should we call them? Just put them with deoxy. So if it is adenosine, they will become deoxyadenosine. Same way, guanosine, it will become deoxyguanosine. If it is a cytidine, it will become deoxycytidine. So there is a difference of putting D. And what will happen in place of uridine? It will become thymidine because it is a methylated in case of a DNA. So uridine and thymidine is the difference. All others are different in terms of just deoxy or oxy group. So whenever we, there's a notation which is given. So if I say PA, that means it is containing phosphorus residue, and A is adenosine. So this becomes a nucleotide. Here, phosphate residue and cytosine, phosphate residue and guanosine. So these residue, since they contain phosphate residue, they will become nucleotide. And you have the term adenosine, guanosine, cytidine, and uridine, right? Uh, the if I write PACG, so all of them, they are denoting a trinucleotide of DNA consisting of a building block of adenylate, A, cytidine, and guanosine. So these are monophosphate, they are linked together. P denotes to the phosphate group. There is nothing much except that whenever you see this, in case in how they are abbreviated, what are the notation, you should be able to understand that. So coming to the nucleotides, these are the nucleotides. DNA, so you can have a very long chain of nucleotides. So DNA is not just 2 nucleotide, 3 nucleotide, or 10 nucleotide. There is huge, you know, poly, which is of a virus such as a papilloma virus, which is normally known for cancer causing, or is a cancer causing organism. It is a long 5100 nucleotide in the length. So there is a huge chain which is present. And not, so when I'm talking about a small virus, I have talked about 5100 nucleotide, which is a very small one compared to human genome. When I talk about the human genome, you will have 3 billion nucleotides, and they are divided among 24 distinct DNA molecules having 22 autosomes, X chromosome, Y chromosome of different sizes. Now looking at some of the nucleotides which have a different function than the one which you have learned. So some of the nucleotides, for example, this one is a 5' ATP molecule. Now this molecule is energy currency. I'll discuss what is the advantage, and what is the sugar residue here is a ribose sugar. And this is a nucleotide known as adenosine triphosphate. This is the GMP molecule. They will be involved in the signal transduction. And if you look at the structure, for example, ACG, so this is adenosine, this is a cytosine, is a guanine, and they are having phosphate residue. So you have one nucleotide here, another nucleotide, third nucleotide linked by phosphodiester bond. So after that, these DNA molecules, they have a organization. And normally what happens that they are having double helical structure. So covalent structure of nucleic acid, that are the one, they can carry information in one form, in the form of sequence of bases along the nucleic acid chain. So suppose I have a nucleic acid chain, this is a nucleic acid chain. So these are the chains, say blue color and red color, they are the one which are polynucleotide chains, and they carry the information. The bases found in nucleic acid to form a specific base pair in such a way that helical structure consists of two strands and they are formed together. Then double helical structure for DNA facilitates the replication of genetic material. You will learn how it happens. Then double helix is stabilized by the hydrogen bond and hydrophobic interaction. I'll tell you what kind of hydrophobic interaction, what kind of hydrogen bond. And this structure was given by James Watson and Francis Crick, which has actually changed our understanding about the molecular biology. So if you want to understand molecular biology, the structure of the DNA became fundamental. And they, based on the X-ray structure, they have given this information. So you get the information about the property of the DNA. So how the DNA are, you have one polynucleotide chain, then there is another polynucleotide chain, and I'll tell you how they are organized. So this is giving you a basic information how the DNA structure looks like. So in this DNA, these rod-like structure, they are the base, they are the one which are the linkage between the two base residue. So suppose this is a chain, this chain will have different type of bases. So suppose here you have presence of A, you have T, thymine, you have G, you have C, and they are linked. So A will connect with the T, and this kind of a hydrogen bond is present. Here T is present, in the other side you have a complementary nucleotide. Here if you have a G, here you will be having C. So we'll talk about how this organization occurs, but you can imagine that one polynucleotide will have certain sequence, the other will have a complementary sequence. So if A is present on this side, T has to be present on the other side. If T is present, A will be here. If G is present here, C will be present there. So that's the way the organization occurs. Now, the bonding between these two bases will be by hydrogen bonds. So there is a hydrogen bonding between the different base residue. And so what will happen? So this chain, they will contain the backbone. That means it will have a sugar residue and a phosphate. Phosphate is present on the outer surface. So why we call the nucleic acid as a nucleic acid? Any one of you? Because they have lot of phosphate residue, and if you look at the pK value of a phosphate group, it leads to the acidic characteristic of the DNA molecule. And other thing which you will learn in this is that what are the features of the Watson DNA Crick model, how the genetic information flows from one generation to another generation, and some information that how this double helical DNA opens up and passes on the information. So I'm stopping at this point, and we will talk further about this. So if you have any question, you can ask. Any query, any question? So if there is no more question, I'm going to sign off, and thank you very much.